So today I'm going to talk about hippocampal coding and the fact that in the hippocampus there are multiple scales of coding, multiple temporal scale, kind of a multiplexing of routing of information, and how this temporal scale can interact with each other. So as I imagine that a number of you will be not familiar with hippocampal coding, what is important to know is that the hippocampus is a brain region which is relevant for us to understand where we are in the space. So for example, it's also very important for memory formation and other cognitive functions, but in this specific task it's important that this linked to our spatial location. So for example, there are place cells. Place cells are units which activate whenever we pass to a specific point in a space. So the specific point is called place field, and each unit might have one or multiple place field, and they reliably activate whenever the animal crosses that specific location. Now, these place cells are active on a time scale of seconds. So this is the behavioral time scale that we find within the hippocampus. However, there is still within the hippocampus a much finer time scale at the millisecond level. So this is the time scale of the theta sequences and phase precession, which I don't have time to go into now. So theta sequences are very organized, precisely coordinated firing of units within the hippocampal network. So here you see 3 place cells with neighboring place field, and you see that at the time scale of a theta sequence, which is approximately 8 Hz, you see these ordered sequences of activation with within the spiking of these 3 units. So this sequence of sharp temporally coordinated activation of unit of the hippocampal network. This is a completely different time scale of coding, which is at the millisecond level. Now, these theta sequences initially were thought to be mainly related to also spatial information, but in the latest year we have been seeing that they can also carry task-relevant information. For example, there are different theta sequences that encode alternative choices that the animal has to evaluate within few milliseconds, or there are theta sequences which encode goal location, because in fact it has been observed that when the animal has a goal-directed behavior, so it's going somewhere, the theta sequence ends with the goal location, even if this goal location is very far from the actual position of the animal. So there is a cognitive manipulation of these theta sequences that this doesn't just reflect spatial information. So this is the introduction. So what we did, so this project was in collaboration with the group of Matt Jones in Bristol University, and in particular in this task, Nadine recorded from these hippocampal units, CA1 units, and overlapped, performing a working memory decision-making task. Now, in this task, there were 6 possible trial types. There were 2 choice trial types in which the animal had to start from this location or from this location, cross the central alley, and then at this point perform a decision whether to turn left or right, depending on whether the animal started the trial from this or this platform. So he had to remember. So at this point there was a memory load that was different for the 2 trial types. These other trial types are instead guided trial types, in which the animal practically doesn't have to be to perform any decision, because when going back, here at this point there is a movable wall that can direct the walking of the animal either in this arm of the maze or in this other. So he has just to go back, but without performing any particular decision. So this was the task. And we were interested in investigating in a supervised manner the coding structure that we could find within the data. Now, in particular, we were interested in finding cell assembly. Now, cell assembly is a mixed term in neuroscience. Initially started with a specific, relative to mark specific engram that we also say, that are units which are activated together and they are wired together to encode a specific behavioral information. But nowadays it's used in a bit looser sense to mark any coordinated activity of a group of units. The only important thing is that this coordinated activity has to be standardized and repeated in a systematic way. So while the single unit has an ambiguous information, so can be active to encode multiple stimuli. The cell assembly, its activation together with other units of the network is univocal. So for each behavioral item, there is a specific assembly, while at the single unit level, we have ambiguity. Now, we did use an algorithm that I developed some years ago to identify this pattern of recurrent activity pattern in the data in an unsupervised manner. And what this algorithm can tell us, if we just simply give the data without any behavioral information, the neural activity data, it can tell us if there are these reoccurring patterns. So we have to find suprachance motifs that reoccur in the data. Which are the units which compose this pattern? What's the pattern of activation of each unit? So here you see very different example possible assembly activation, either synchronous or sequential, either very precise temporal coordination or a more loose rate-based coordination. Why I make this distinction between the time scale? Because this algorithm specifically can tell us also the degree of coordination of the spiking of this unit. So it can tell us whether we have a situation like this in which the spiking activity is coordinated at the millisecond level, or if we have a situation like this in which what is relevant in the coordination is the average firing rate of the unit, but the single spike we can jitter it, and we don't lose any information. So we have this tool, machine learning tool, we can say, and we apply to our data from the group of measures. And what we found, as we expected, we found a lot of assembly, a lot of coordinated activity in this network. We found that the same unit could take part in multiple assembly in different moments of time. And interestingly, we found that indeed the time scale of coordination, so the time scale of coding for this region was diverse. So we found two major time scales. One that was on the time scale of seconds in this coordination, and one that was of the time scale of milliseconds. So the coordination of the unit firing within the assemblies at a millisecond precision. So, and we call this rate and spike assemblies, these two broader groups. These are what this activity pattern look like. So this is an example of the rate assemblies. You see here the time scale is much larger than this for the spike assemblies. And looking at the activation also during the maze of this assembly pattern, we can see that the rate assembly would match the coordination between different place fields of the hippocampus, of different place cells of the hippocampus. On the other hand, these spike assemblies, which have a much finer time scale, as you can see here already from the time axis, they are compatible, at least, with the activation of theta sequences. And here I plotted the local field potential. So if these are theta sequences, they should be coordinated with the theta rhythm of the local field potential. And if you squint, you can see already that there is a bit of coordination. Now, to study this coordination in a bit more formal way, we look at the phase preference of this unit. We know that the hippocampal units show, in general, tend to show a phase preference with respect to the theta rhythm. What you can see here, however, is that, for example, if we take a unit and we sort its spike from those that have been fired within the cell assembly and those that have been fired outside of the assembly configuration, we see that when we are within the cell assembly, the phase preference of this spiking activity with respect to the theta rhythm is much narrower than when I consider the full distribution of spikes of the same unit. So the same unit has a narrower phase preference when active within the assembly. Then we ask, okay, but this unit could take part in multiple assemblies. So is this phase preference a unit-specific feature or is an assembly-specific feature? What we noticed was that in a large majority of cases, a large fraction of cases, a unit, which, for example, in this case, unit 2, is part together with unit 3 in assembly 1A and together with unit 1 in assembly B. So these are active in different moments of the trial. The phase preference was different for the same unit when active in one assembly or the other. So it looked like the phase preference of the unit was assembly-based rather than a characteristic of the unit itself. this narrower preference of phase for the unit within the assembly was also associated to a narrower and more specific information carried by the unit. For example, here you can see a unit, unit 2, which is part with unit 1 in assembly A and part of unit 3, and together with assembly 3 is part of unit B, of assembly B. Now, this unit, we see I linearize the activity of the unit on the path, we see is a bit active during left guided trials, a bit during left choice trial, a bit in right guided trials, but together in the assembly activation, the assembly has a much clearer and encoding property. So here, together with unit 1, we select and highlight the activation for left guided trial. And indeed, more in general, we saw that for the same unit, in general, it took part to different, when the same unit had multiple place fields, in different place fields, the unit activates together with different assemblies. And in some cases like this, even within the same place field, for different trial type, it activates together in coordination with different other group of neurons. So this has led to do another test, because if different place fields match with the activation of different assembly, we said, okay, let's just forget about the assembly for a second, and let's just divide the spike of the unit, of the same unit, by place field. So this is the activation of the unit, of one unit, for example, in a maze, and we cluster its spike in the 4 different place fields that he has, and we said, do we see a different phase preference? Do we see a phase coding in this group of spike? Well, yes. So here are 2 different examples. These are 2 units with different place field, and you see here the histogram of the phase preference. And clearly there is a shift in preferred phase of firing when the unit is active in a place field or the other. So there is a kind of phase coding. But as we saw that assembly could also differentiate the activity of the unit within the same place field, we ask ourselves, for the same place field, if I split the spike and I divide them and I look at the phase for different trial type, is it going to be a different phase? Well, this histogram, for example, split in this different modulation of phase. So it looked like that clearly this unit had a different phase preference when active in different trial type, even within the same rotation. And indeed, we could decode the trial type exclusively based on the phase. So another interesting thing we noticed is that this phase coding was more prominent when we were comparing trials which needed to be different for the execution of the task. So we noticed a different phase coding, for example, for different choice trials, so when the animal had to remember if it's and distinguish between left and right, or whether choice between choice and guided trials. So that were 2 completely different types of trial. And we saw little differentiation instead when we compare different guided trials. So trials in which the animal didn't have to remember anything and just follow the maze as such. So how can the activation of different cell assembly, which are something at the rate scale, lead to the change in phase at the single unit level? Well, we know from the literature for studies on phase precession, which I didn't mention for time reasons, that if we modulate the internal depolarization of an hippocampal unit when there is an oscillatory field like the theta rhythm, we can modulate the phase of the unit. So we thought, okay, maybe different assembly activate different standardized depolarization for the unit, which then cause the different phase preferences. Now, in our experiment, we didn't have access to the internal depolarization of the unit, the membrane potential, but what we did, we looked at the instantaneous firing rate of the unit, so how it's modulated in time, and we saw that when we add a different phase preference, we also had, not in absolutely all cases, but in the majority of cases, we had a different rate preference. So to a different phase, it also corresponds a different instantaneous rate of the unit. And we found these results when we compare different place field of the same unit, or when we compare within the same place field of the same unit different trial type, or when we compare the activation of the unit in different assemblies, different phases, different rate. Okay, Then this led us to formulate an hypothesis, because it can be that when we have different cognitive demands, when we need to remember I was coming from left to right, when I am in different contexts, even though they are not clearly signaled by my environment, I will give to the unit a standardized degree of depolarization, and I will produce a phase preference that is given by the assembly, because the important thing is that the assembly is a stereotypized structure. So the depolarization also will be stereotypized whenever the assembly is active. And this will lead to a stereotypized phase preference. I have to go very fast. We did the model. So to test if this was reasonable as a hypothesis, we did a model in which the same 3 units were active in different assemblies. And this assembly, the activation of these assemblies were marked with they were causing a different degree of depolarization of the units in the network. So assembly A, for example, activate very little this green unit, but activated a lot, but assembly B activated a lot the green unit. So the 3 units are in 2 different assemblies. They are still in the assembly, but the assembly, because are globally at the brain level, they are recruiting different inputs that will cause a different depolarization. And we notice that this degree of depolarization, that is the only things that we change between the 2 assemblies, led to a different phase preference of firing of the unit. So this is, at least in the model, is a causal link given by the depolarization. And interestingly, led to different data sequences. The generation, instantaneous generation without wiring need of different data sequences. We look in the data whether we actually did that at this point, different data sequences. And indeed, we found that the lag between 2 units, which had overlapping phase fields, changed in a fraction of unit systematically for one trial type or the other trial type. So when one assembly was active or the other, the lag of activation, so their relative position in the data sequence, changed because the data sequence was shuffled. Another interesting observation was that the last units firing in the data sequence is the one that is least depolarized in our model. And this is interesting because I told you at the beginning of the talk that when we have goal-directed behavior, my goal direction, the location of my goal, fires always at the end of the data sequence. So we can imagine that when I have an objective, the cell that signals my objective is slightly depolarized throughout the trial, because that's my objective. If this would be the case, and has to be tested experimentally, then if this would be the case, this would put the activation of the firing of that cells automatically at the end of data sequences, as has been shown by experimental results. I skipped the conclusion because I'm late, but it's just a recap. So the different assembly activation lead to different stereotypical phase preference of hippocampal units, which lead to a flexible mechanism to generate different data sequences in my network. Why this is important? Because the data sequences bring something that is at the time scale of seconds, so at our behavioral time scale, at a much finer time scale that is compatible with plasticity. So this could be a mechanism to store information that occurs at the behavioral level at the fine time scale of synaptic plasticity. And indeed, the data sequences have been linked to replay, sleep replace, and the storage of them. So this would be a neat way to store more complex information within our network in a flexible way. I want to thank all my collaborators for this project. In this case, the major job was done by Nadine, which performed all the experiments within the group of Matt Jones, and also with the collaboration of Daniel Dustemint. And I always want to thank my group, which was not directly involved in this project, but is very active in all of my current projects. So thank you very much.
So it has always surprised me in the results of Nahum Ulanovsky when he looks at bats. They don't have theta, but they have place cells. So how would this encoding work if you didn't have theta?
So I would expect not to have theta sequences, and this might affect plasticity, but of course theta sequences are not the only way to store memory in the network. They are just a tool to push it a bit, to highlight it. So it's a way that favors it, but it not necessarily has been linked. I mean, there are other ways to store memory in the network. And there has been an experiment in which, even in rats, they managed to maintain the rate modulation but destroy theta sequences. And what has been shown is that they were affecting the replays by night. So the destruction of theta sequences was affecting the replay by night. So I'm sure this rat eventually learned something, but the link between replays and the replays then are linked to memory consolidation was dependent on the theta sequences. So I don't know how bats do.
Other questions? So I have a question. So one thing that I have to confess I never understood properly was that, so theta are presented as almost an external frame of reference. So my question is always, I mean, whether they come from spiking populations, LFP, doesn't matter. So how is it possible that the presence of sequence of spikes doesn't cause any, I don't know, phase shift, slight change in frequency in theta? So how comes that theta are this universal frame of reference in which you place your patterns?
So theta is modulating the activity of the hippocampal unit through somatic inhibition in general. It's thought, and it is not locally generated, so the spike activity of the local unit is not.
But there's no loop coming back.
Probably. I have to say that I'm not such an expert in theta per se. I'm more on the methodological approach, but yes, per se within the CA1, it's not generated within the network.
So it's really a clock that doesn't depend on the local activity
of CA1.
Of CA1. Okay. Other questions? If not, let's end the speaker again.